Good day everyone! For today's report, we will continue to study on two more genus from phylum Metamonada, which was previously introduced by another group. And we will also take a look at another phylum, which is phylum Ciliophora. Let's begin with the genus Giardia. Giardia is a protozoan parasite that belongs to class Metamonada, order Diplomonadidae, family Hexamitidae, and subfamily Giardinae. Giardia is one of the most widespread intestinal protozoan parasites. It colonizes and reproduces in the small intestines of several vertebrates, causing giardiasis, a diarrheal disease that we will further discuss in a while. Giardia is found on surfaces or in soil, food, or water that has been contaminated with feces from infected people or animals. It is characterized by the absence of mitochondria, although it has retained a reduced organelle of mitochondrial origin called the mitosome. As seen from the figure on this slide, Giardia has 8 flagella that they use for motility to find suitable sites for attachment to the intestinal villi of their host. The 8 flagella are organized as 4 pairs: the anterior, caudal, posterior lateral, and ventral flagella. As for its vector, the role of indirect transmission mechanisms has not been extensively studied. However, in a study conducted by Doyes et al. In 2000, it showed that Giardia cysts are most commonly found in flies, arthropods, and cockroaches. Flies, specifically, were described to be important transport vectors between feces and food, therefore playing a significant role in the transmission of giardiasis. Hosts of Giardia include humans, wild animals, and domestic animals. A study by Adam in 2001 showed that each of the 6 Giardia species has its own specific host. Amphibians serve as the main host of Giardia agilis, rodents for Giardia muris, mammals for Giardia duodenalis, cetacine birds or parrots for Giardia psittaci, herons for Giardia rda, and voles and muskrats for Giardia microti. People are considered to be the most important reservoir hosts for human giardiasis, a disease caused by Giardia duodenalis. Giardia duodenalis is the only Giardia species known to cause illness in humans. While there are 40 species of Giardia that have been described, many of them have been presumably synonymous to one another. Presently, as said in the earlier slide, 6 distinct species are recognized, as presented in the pictures below. Scientists differentiate between species through morphology and morphometrics, host specificity, isoenzyme, PCR, and antigenic analysis and molecular methods such as karyotyping.
Giardia causes the diarrheal disease giardiasis. Since Giardia can be found on surfaces or in soil, food, or water that has been contaminated with feces, giardiasis can easily infect many people and can be found worldwide. Once a person or animal has been infected with Giardia, the parasite lives in the intestines and is passed in stool. And once outside the body, Giardia can sometimes survive for weeks or even months. The life cycle of Giardia starts with the ingestion of Giardia cysts from contaminated water, food, or by the fecal oral route. Then, in the small intestine, excistation releases trophozoites, which is the activated feeding stage in the life cycle of Giardia. These trophozoites multiply by longitudinal binary fission, where they remain in the lumen of the proximal small bowel, where they can be free or attached to the mucosa by a ventral sucking disc. Encystation then occurs as the parasites transit towards the colon. The cyst is the stage found most commonly in stool. Afterwards, they are free to infect more people. Giardiasis occurs worldwide. Oftentimes, the most high-risk groups are infants, young children, international adoptees, travelers, etc. It is very common in areas with poor sanitary conditions and limited water treatment facilities. In resource-limited settings, the prevalence of giardiasis has been reported to be as high as 20 to 40%. It is the third most common agent of diarrheal disease in children below 5, and more than 300 million cases are reported annually. However, many individuals with Giardia in stool samples are asymptomatics, so you may be carrying and spreading Giardia without even knowing it. Symptoms of this disease includes the following: diarrhea, gas, foul-smelling greasy poop that can float, stomach cramps or pain, upset stomach or nausea, and dehydration. It usually begins by having 2 to 5 loose stools per day and progressively increasing fatigue. Less common symptoms include fever, itchy skin, hives, and swelling of the eyes and joints. Over time, giardiasis can also cause weight loss and can keep the body from absorbing nutrients that it needs, like fat, lactose, vitamin A, and vitamin B12. Though, as mentioned, some people with giardiasis may have no symptoms at all. Medications such as metronidazole, tinidazole, and nitazoxanide are prescribed if the symptoms are severe. But usually, many people who do have problems often get better on their own in a few weeks. It's also often advised to drink plenty of fluids to prevent dehydration, especially for infants and pregnant women. Because of how easy Giardia is able to spread from one person to another, preventive measures are required. One of the most important acts would be practicing good hand hygiene, especially since Giardia can often be found in soil. Another thing would be to avoid water or food that might have been contaminated. Do not drink untreated water from lakes, rivers, springs, ponds, streams, or shallow wells. Do not eat fruit and vegetables washed in water that might be contaminated. Another preventive measure would be to practice safe sex. Since Giardia may be in poop for several weeks, wait to have sex for several weeks after you or your partner no longer have diarrhea. Last and most importantly would be to clean and disinfect yourself and your surroundings.
Next is genus Hexamita. Hexamita is an intestinal flagellated protozoan parasite that belongs to the class Metamonada, order Diplomonadida, and family Hexamitidae. Hexamita is a small, approximately 3 to 18 microns intestinal parasite. The members of this genus have a pyriform body with 2 nuclei, 6 anterior and 2 posterior flagella, and 2 axostyle. It is commonly found in the intestinal lumen of freshwater fish. It has a double set of organelles but lacks mitochondria and Golgi, thus it has a primitive metabolism. Its mode of transmission is direct, thus there are no vectors. Transmission occurs via fecal-oral route contamination and ingestion of contaminated water and food. Studies have shown that it has two morphologies. First is the trophozoite form at its infectious stage, in which it is oval-shaped and bilaterally symmetrical with 8 flagella, 4 anterior, 2 anterolateral, and 2 posterior flagella. Moreover, it is also binucleate with a prominent nucleus and is approximately 6 to 12 micrometers long. It also exhibited a cyst morph but is only rarely formed. The most important species is Hexamita melayugridis, which occurs in Turkey. Hexamita melayugridis is a protozoan parasite of turkeys, pheasants, pigeons, and some game birds. It is transmitted by feces, fomites, and carriers. It occurs in the duodenum and intestine of young turkey and causes infectious gastroenteritis. Other members are Hexamita columbae, Hexamita muris, Hexamita piteki, Hexamita salmonis, and Hexamita trutei. Members of the genus Hexamita parasitize wild, farmed and aquarium freshwater fish and amphibians across the world. In cold and temperate waters, many fish families are potential hosts. Hexatima salmonis most commonly parasitizes salmon species. Moreover, Hexatima columbae live in the intestines of pigeons. Hexatima muris and Hexatima piteki live in the intestines of mammals, and Hexatima salmonis and Hexatima trutae live in the intestines of fish. Hexamitiasis is caused by intestinal flagellated protozoa that attacks the digestive system and the lower intestines of infected fish. These are likely present at low levels in the intestines of many aquarium fish. It is only when the fish is weakened in some way that the parasites can multiply rapidly and spread into the rest of the fish body. And when the parasites become established in the other internal organs, the fish dies. Fish infected with Hexamita commonly produce white, stringy feces, and their color becomes paler than normal. The lesions are caused by Hexamita parasites that have migrated into the sensory pores on the head, which leads to the decay of the surrounding tissue. Because of the damage to the epidermis, secondary infections can easily occur. On the other hand, it is uncertain that all cases of head and lateral line erosion disease in fish are caused by Hexamita parasites.
Like mentioned a while ago, the genus Hexamita has a direct life cycle in which the parasite is transmitted directly to the host without the act of vectors or intermediate host from other species. This is done through the feeding of the host or the organism who ingested a contaminated food or lives within a contaminated environment. Flagellated Hexamita asexually reproduces through longitudinal binary fission. When they are ingested, they invade the intestinal mucosa of the host, which results to what we know now of as infection, or the head bumps and other clinical manifestations. Transmission most likely occurs through fecal or oral transmission. Hexamita often invades fishes within the cichlid genera, which includes angelfishes, discus, oscars, and the like. They can also infect birds and mammals. It was mentioned a while ago how Hexamita is transmitted directly through ingesting, thus the environment plays a huge factor. Poorly sustained environments often cause the thriving of Hexamita. For cichlids, extreme population or crowding within a certain place such as an aquarium and poor water quality influence the prosperity of the genera. Other environmental factors include shipping and handling when the animals are transported and contained in a substandard environmental condition and improper nutrition. Infected animals also increases its vulnerability when other parasites compromise the immune response. As Hexamita invades the body, the immunity of the host is compromised. The host refuses the food, leading to weight loss and poor body condition due to the lack of nutrients. Head bumps and holes will be the palpable signs that the organism has been infected. Other clinical manifestations include the inflation of the abdomen and pale-colored feces. Specific for severely infected angelfishes, they would often lie horizontally on the surface. The Xamita's damage affects a great deal for the host's life. As such, treatment and preventive control is needed. As mentioned, poor environmental conditions influence the prosperity of Hexamita, which is why in order to kill or prevent them, it is highly suggested that the environment must be properly restored and cleaned. Aquatic environments such as aquariums need biofilters or just water being cleaned for a couple of times. For infested animals, metronidazole is the prescribed drug taken orally or through bath immersion when the organism cannot ingest anything. Other preventive measures include sustaining a proper and healthy environment for the organisms and good monitoring. Let us first have a brief background on phylum Ciliophora. Phylum Ciliophora or Ciliata constitutes a homogeneous group of highly differentiated unicellular organisms. They are primarily distinguished by the presence of cilia, which are short hair-like structures that they mainly use for locomotion and eating. These cilia structures are also arranged in lines called kinetes, which provide the underlying structure of flagellar roots around ciliates, and the arrangement of kinetes is also an important morphological feature for ciliate identification. Ciliates are typically larger than most protozoa, with sizes ranging from 10 to 3,000 micrometers, and approximately 8,000 species have been described. Aside from the presence of cilia, another distinguishing character of ciliates is nuclear dimorphism, in which each cell has two kinds of nuclei, one macronucleus and one micronucleus. The macronucleus is responsible for controlling RNA synthesis for cellular regulation, while the micronucleus is exchanged during conjugation with another cell when ciliates reproduce sexually. Most ciliates are motile and have adaptations that allow them to move into the plankton, but some species were also described to be benthic. Some species use stalks or a gelatinous matrix to attach to substrates, and some are found attached to detritus. There are many genus belonging to phylum Ciliophora, one of which is the genus Paramecium, which is an important model organism. Today, we will be focusing on its representative genus Balantidium.
We will now proceed to another parasite, which is the Balantidium. Balantidium is a genus of ciliates. It contains the parasitic species Balantidium coli, the only known cause of balantidiasis. It belongs under the class Lithostomatea, order Vestibuliferida, family Balantididae, and the genus Balantidium. So, the Balantidium general characteristics. Balantidium is the largest protozoan and the only ciliate known to parasitize humans. Its distribution is worldwide. The definitive hosts are pigs and rats are important sources of infection for human beings. Pigs are the main animal reservoir, and it's also reported in dogs, cows, horses, rodents, and non-human primates. There is also a man-to-man transmission is rare, but possible. The intermediate host, there are no known intermediate hosts or vectors. The Balantidium mode of transmission. Cysts, which are the infective stage, are responsible for transmission of balantidiasis through ingestion of contaminated food or water through the oral fecal route. Water is a vehicle for most cases of balantidiasis. The site of infection is the cecum and colon. Its virulence factor is the hyaluronidase, which helps to penetrate the intestinal mucosa, and its excitation occurs in the small intestine. The figure below shows the differences between the cyst versus the trophozoite. Cysts and trophozoites are two stages of the life cycle of protozoans. The cyst is the infective stage of Balantidium, while the excitation produces a trophozoite from the cyst stage, and a single trophozoite forms from each cyst. We will now describe the trophozoite. The trophozoite is the active reproductive stage of the protozoans that feed on the host. It is found in active stage of disease, the dysenteric stool, and its invasive form shape is oval. Its size is 30 to 300 micrometer long times 30 to 100 micrometer in breadth. The whole body is covered with a row of tiny delicate cilia, which is an organ of locomotion. Cilia is present near the mouth part, and the longer cilia are called adoral cilia. The anterior end, narrow, bears a groove, which is the peristome that leads to a mouth, which is the cytostome, followed by a short funnel-shaped gullet, which is the cytopharynx extending up to 1/3 of the body. The posterior end is broad and round, bears an excretory opening called the cytopyge. It has no anus, and the cytoplasm is an outer clear ectoplasm and the inner granular endoplasm. One or two contractile vacuoles lie side by side or one above the other, maintaining the proper osmotic pressure inside the cell. It has also numerous food vacuoles which contains food particles like debris from host gut, bacteria, starch grains, fats, droplets, and occasional RBCs, where digestion of food particles take place. The endoplasm contains two nuclei, one macronucleus, which is a large kidney-shaped macronucleus in the center and responsible for the cytoplasmic activities, and also the micronucleus, which is a small vesicular nucleus which lies in the notch of the macronucleus and responsible for reproductive process. And finally, we have the cyst. The cyst is a dormant stage of the protozoans that resist unfavorable environmental conditions. Its shape is round, its size is 40 to 60 micrometer, it is immobile and dominant, it is surrounded by a thick transparent cyst walls which allow the cyst to resist degradation in the acidic environment of the stomach in the basic environment of the small intestine. It contains two nucleus, which is the macronucleus and micronucleus and the vacuoles. Cilia is seen in younger cysts but is absorbed on maturity, thus the movement ceases. It's an infective stage of animals and it has a non-reproductive stage.
Moving on to the disease, Balantidium coli is the only ciliate and the largest protozoan parasite of human. The organism appears to be primarily a parasite of pigs, with strains adapted to various other hosts, including humans, rodents, and other primates. Balantidiasis is the disease caused by Balantidium coli, and balantidiasis in humans is most common in the Philippines, but can be also found anywhere in the world, be it in the tropics or temperate areas, especially among those who are in close contact with swine. Generally, the disease is considered rare and occurs in less than 1% of the human population. However, pig infection rate is high with overall estimated prevalence of 100%. For their life cycle, the host can acquire the cysts or the dormant stage of the parasite through the fecal oral route by contaminated food and water. Following ingestion, excistation occurs in the small intestine to produce the trophozoites or the growing stage, which will colonize the large intestine. The trophozoites will then replicate by binary fission, then it will undergo encystation to produce infective cysts. Some trophozoites invade the wall of the colon and multiply, some will return to the lumen and disintegrate. Then the mature cysts are passed with feces. In pigs, if the feces do not dry out immediately, the cyst can remain alive for weeks. Next slide, shown in the figure below. On the left side is the Balantidium coli cysts or the dormant stage, and on the right side is the trophozoite or the active stage found in fecal matter. Now for the clinical manifestation, most cases of balantidiasis infections often disappear spontaneously in healthy persons, or they can become asymptomatic, making them a carrier. Sometimes, symptoms are present, which include abdominal discomforts, diarrhea, and some secondary infections. Those secondary infections could be fatal to an immunocompromised person. For the treatment, control, and prevention, drugs like tetracycline, metronidazole, and iodoquinol are used to treat infections of Balantidium coli. Prevention and control measures include proper hygiene, like washing hands with soap and warm water after handling anything fecal matter related, especially for those who work with pigs, and thoroughly washing fruits and vegetables with clean water and then prepare when preparing or eating them is also advised. And that concludes our report. These are our references. Thank you.